Hello everyone, my name is Joey. Today I would like to talk about the anatomy and the enzymology of fat absorption. This is my agenda today. I would go through this order. On the left, I also show the picture of the small intestine, which is the primary places for the absorption within human body. I will go through from the bile salts, lipase enzyme, micelles, and phospholipase and fat absorption process. The first part is bile salts. Nearly 19% of our fat calories come from triglycerides because they are the primary form of lipids in both plants and animals. Fat digestion is complicated by the fact that most lipids are not particularly water-soluble. As a result, the aqueous chemical leaving the stomach contains a close atomization of large fat droplets, which have less surface area than smaller particles. To increase the surface area available for enzymatic fat digestion, the liver secretes bile salts into the small intestine. Bile salts can help break down the coarse emulsion into smaller, more stable particles that clump together when digestion exposes only surface molecules into lipase enzymes. Bile salts secreted into small intestine to physically emulsify lipids. They separate lipid clump and increase surface area. The enzymatic fat digestion is carried out by lipases, which is an enzyme that removes two fatty acids from each triglyceride molecule. The result is one monoglyceride and two free fatty acids. The bile salt coating of the intestinal emulsion complicates digestion. However, because lipase is unable to penetrate the bile salts, for this reason, fat digestion also requires calypsin, a protein cofactor secreted by the pancreas. Calypsin displays some bile salts, allowing lipase access to fat inside bile salts coating. Now, let me move to the micelles and phospholipids, which are the two most commonly terminology we can heard about this in our normal life. Phospholipids are digested by pancreatic phospholipase. Free cholesterol is not digested and is absorbed intact. As enzymatic and mechanical digestion proceeds, fatty acids, bile salts, mono and diglycerides, phospholipid and cholesterol coalesces to form small disc-shaped micelles. Micelles then enter the unstirred aqueous layer at the edge of the brush order. Now this is our final part about fat absorption process. Here shows a picture about fat digestion and absorption process. The digestion is from the number 1 to number 3B. But our presentation today mainly focus about the later part, fat absorption, which is indicated by the number 4 and the number 5. I have already mentioned many processes before. Additionally, I have to say that lipophilic fats such as fatty acids, monoglycerides are absorbed primarily by simple diffusion. They move out of their micelles and diffuse across the interstitial membrane into the cell. Once the monoglycerides and fatty acids are inside the interstitials, they move to the smooth endoplasmic reticulum, where they can recombine into triglycerides. And the triglycerides then join cholesterol and the triglyceride and proteins to form large droplets called chymocromosomes. Because of their size, chymocromosomes must be packaged into secreted vehicles by the Golgi. The chymocromosomes then leave the cell by exocytosis. The large size of chymocromosomes also prevents them from crossing the basement membrane of capillaries. Instead, chylomicrosomes are absorbed into lacteals, the lymph vessel of the body. Chymocosons pass through the lymphatic system and finally enter the venous blood just before it flows into the right side of the heart. Some shorter fatty acids, which is 10 or fewer carbons, are not assembled into the chylomicrons. This fatty acid can therefore cross the capillary basement membrane and go directly into the blood. So here is our presentation today. This is the reference I used in our presentation. If you have any questions, please feel free to contact me. Thank you.